Hey, thanks, Peter. My name is Mahmoud Ahmed, and I'm a postdoc at Gyeongsang National University in South Korea. So my talk is about a database and a Shiny application for cancer cell-specific perturbations of biological networks. And in this work, I'm integrating data from causal biological networks, which I will be talking about later, and gene expression data. And the relevance of this talk to today's audience, or the bioconductor audience, is that I'm using a lot of bioconductor packages to do the heavy lifting, and then integrate this with other R packages to do interactive graphs and interactive Shiny application. So this is a brief outline of the presentation, and I will start by just explaining what is a by two introductory slides. The first of which is about what biological language expression is. So the biological expression language, or BEL for short, is a way to code biological interactions into a standard format, so that you can take the knowledge from the literature and you can transfer it between studies in a standard way, and then you can perform computation on it. And the structure of this language is very simple. You have two entities. One of them would be a subject, one of them would be an object, and there is a relationship between them. And there are certain codes that you can use to indicate the direction of the interaction, and you can indicate what function these entities, or what function these entities perform on each other. The second of which, the second introductory slide, is about reverse reasoning. And the idea here is that we take findings from the literature and then integrate these with gene expression data first to, or for any data on these entities in the graphs, to first validate that these interactions are valid. And then you can also do some inference on these networks. So the particular approach that I'm using in this in this work is if you have a network and this network code interactions between biological entities, for example, a transcription factor and a gene, and downstream from this gene there are other transcripts or other genes that change their function based on the node upstream. Then if you can code these interactions into the standard way of the biological expression language, and you can form some sort of expectation. So for example, a transcription factor induces a gene, and this gene codes for a protein that itself has two downstream effectors. And then for example, it inhibits one and induces the other. So if you have this form of expectation, and then you can treat drugs, or you can do gene manipulation and see which of these manipulations result in perturbing the interaction that you're interested in, by looking at the downstream effects of, for example, the changes in gene expression, and see which one matches the expectation that you had based on the literature knowledge that we form in a network. So I'm using two types of data in this work. The first one is gene expression profiles from cell lines treated with different drugs, and this data set comes from the LINPS project. And then I'm using several causal biological networks that come from a project with the same name, and these biological networks are two-layer networks that code interactions compiled from the literature for important cellular functions such as apoptosis and cell cycle and so on. So the workflow of this database is that we start first by using the gene expression data and compare the drug treatment to the control cell lines using lemma, and we get fold change and p-values, and then use this data, use this output to integrate it with the biological networks that we have in a package called MPA, and then we get some sort of we can infer the activity of the nodes upstream from the changes in gene expression in the nodes downstream. And then I will package this output into a database and make this database available in an interactive Shiny app. So the scoring of these networks, the second step of the workflow that I just showed, assumes that the function of an entity, such as a transcription factor, is reflected in the downstream gene expression, or the gene expression of the downstream nodes. So we start by constructing a network of two layers that describe a particular pathway, such as cell cycle or the apoptosis. And then those two layers are a functional layer that encodes the causal relationships between the entities, in this case, gene products or genes or proteins, and a transcription layer of all the nodes downstream from this functional layer. And then using this network perturbation amplitude method, or known for short as MPA, to model the perturbation of the nodes in a biological network as the change in the gene expression from the downstream nodes. This analysis was applied using an R package called NPA. So very quickly here, the calculation of the MPA is based on a constraint optimization problem. So we have differential values by we calculate differential values based on the difference in the gene expression of all the nodes that we're interested in between the treatment and the control conditions, and these values should be close to each other or smooth, and then it should also equal the observed fold change in the transcript layer. So these differential values are calculated by solving this problem, and then the MPA score, or the network perturbation amplitude, would be the sum over the edges of the functional layer. So the interpretation of the scores that we get here is the MPA itself is the MPA score is the sum of the differential values of all the edges in the functional layer. And this amplitude indicates how much or the magnitude of perturbation that a particular condition induces on the network as a whole. And then we can extract or take apart the nodes, the leading nodes that contribute the most to the differential values. And then we can also, if we have multiple nodes, multiple networks in the same calculation, we can calculate something called the impact factor, or the biological impact factor, based on the weighted sum of the scores of the significantly perturbed network. So the structure of the database is like that. So we have, after pre-computing the scores for the 8 networks that I have, and for the about 2,000 drugs in about 70 cell lines, the output is encoded as tables in a SQLite database. And then this database has an interface input panels, and then it also has outputs in tabular and graphical formats. The source code for this project is available here. So I put the link to the slides in the Slack channel so you can check this out. So the Bioconductor packages that I'm using in this work are the jQuery package. This is to download the data from the gene expression omnibus, and the Homo sapien organism package, and this is to encode between different identifiers. Of course, the summarized experiment, which contains the gene expression data, and the LIMA package to perform the differential expression analysis, and the MPA to calculate the perturbation scores. So this is a quick use case. In this use case, I'm looking at the tyrosine kinase inhibitors that target a fusion oncogene called ABL-PCR kinase, and this is really important in chronic myelogenous leukemia. So the first drug to be developed in this class is imatinib, and there are in the literature there are a lot of discussion and a lot of work on what the secondary mechanisms or the alternative mechanisms and contexts where this class of important drugs work. And here I'm trying to see for the ABL kinase inhibitors what other cell lines it could work on, and what other gene products that it could target. So the first of the input panels in the Shiny interactive interface is to choose a cellular context, either by tissue or by choosing the cell line name directly. And then you can select the name of the group of drugs that you're interested in. In this case, most of the drugs are encoded by known mechanism of action. And then you can choose what kind of network you're interested in. In this case, this is a family of networks consists of two models for cell proliferation, including the cell cycle and the JAK-STAT pathway. And the output is produced in this format. The first of the output in panel A is the biological impact factor, and this shows us on the two networks for each cell line what's the effect of the drug. And usually these values indicate how important these drugs in different cell lines. And then in P, it shows the effect of the each of the 5 drugs on the cell cycle in one particular cell line. And you can see that one of the 5 drugs is particularly has a higher magnitude perturbation in this network. And then you can also see which nodes in the network each drug targets more specifically. And then there is another way to visualize this output by using graphs. And these graphs encode the gene products or the entities as nodes and the interactions between them as edges. So in this case, I'm selecting this particular drug in this particular cell line and overlaying the effect of the drug in terms of inhibition or in terms of induction or repression by color. So and then you can zoom in and see which part of the network is more important by resizing the nodes to the magnitude of the effect. So the causal networks encode nodes as directed encode nodes and directed edges between the entities, and these can be extracted from the literature or from databases. And then the MPA method uses gene expression of all the nodes to connect the entities and infer activation or inhibition. So considering those two facts, we can determine if a particular node is activated or inhibited or unchanged to produce a specific function. In the use case that I showed, one way of phrasing the finding is that we see that the cell cycle network is perturbed or arrested in response to, most significantly, this drug through its negative regulation of the two transcription factors through retinoblastoma B1. So the effect I'm talking about here is the induction of this transcription factor and then producing the inhibition of those 3 transcription factors. So the limitation of this work is that it's only valid insofar as the underlying data, meaning the causal links and the gene expression data are you can trust them. And then it only reflects if this data or these causal links and the gene expression data reflects the true biology, you can easily get a novel mechanism. And then at the moment, it's not possible to create customized scores for networks or datasets. So all these scores are pre-computed because this analysis is very resource intensive. And for this exact reason, we also couldn't compare, like couldn't calculate the relative scores on all the networks and all the cell lines, but we had to divide them into groups. And then this computation is applied to the cell lines independently again, because the data size is very large for the resources that I have. So I would like to thank you, and if you have any questions, please let me know in this lecture.
Hi, Mahmoud. I have a question. Is there a vignette for LINPS workflow on slide 7, on your slide 7?
Is there a vignette? Well, I tried as much as I could to document the workflow, but it's not as of yet a Bioconductor package. So if you visit the link, I'm sure there might be some useful comments, and if there is anything I could help with, I'll be happy to.
Is there any other questions? Another question, is this examine the drug effect, or are you check the drug effect or with some experiment or something?
Yeah, so the LINPS project have different cell lines, and they treat them with a panel of drugs. and then they check the gene expression on all genes. So the answer to the question is yes. So there is only drug effect perturbations here, or drug perturbation pre-computed scores here.
Thanks. Is there any other questions? It seems there is no questions. So
Yeah, that's all right. I'm sure we can all have
thank
a
you,
break
Mahmoud.
now. Yeah,
Thank you,
no worries.